Adaptive immune system, right? So I'm back here to continue with immunity. This next part is quite fascinating, quite interesting, quite intricate. I think you'll enjoy this section. We've looked at kind of the innate system, something that we all share. We all have that similar surface barriers, similar proteins, complement that enable us to stay healthy. Well, now we're going to shift to this division of the immune system, the adaptive defenses, right? Adaptive defenses can also be divided into two big categories. Humoral immunity, and I don't necessarily think that's the best phrase or term, humoral. When I think of humor, think of comedy, that kind of stuff, funny stuff. Humoral is basically an old English term that means like the different humors of the body, the different liquids, the different chemicals, the different fluids of the body. So it kind of pertains to blood itself. We say that, well, it pertains to the actual white blood cells here. I'll elaborate a little bit more on that. But we're going to have this idea of B cells and antibodies for humoral immunity, and then the thymus immunocompetent cells that we call the T cells, T lymphocytes. So let's jump into our adaptive defense system. So the adaptive immune system is the specific defense system. It's adaptive, right? So it's unique to you and only you. Even if you have a twin brother or twin sister, yeah, they're going to be very, very similar, but your immune system has adapted to you specifically and no one else, right? So that's that adaptive system. It protects against infectious agents and normal and abnormal body cells. We can go back and draw help from the innate system, from inflammation again, from complement again. So part of the immune system, right? We have the two branches, innate and adaptive, but those two branches are interlinked as well. So first of all, adaptive defense, right? Adaptive defense has to have two qualities to qualify as an adaptive defense, right? One, it has to be systemic. Let's say that you were unfortunate enough as a kid, you stepped on that rusty nail. Not very fun, right? Hurts. So when we get immunity, say we got exposed to the little tetanus bacteria, right? The Clostridium bacteria. So we got exposed to that on our left foot per se, right? But let's say later in life, I get scratched on my right arm. Oh man, that little tetanus bacteria got into my system again. Well, immunity, adaptive immunity is systemic. So it doesn't matter where you get that shot, where you got the rusty nail, where we get scratched. That immunity is conveyed throughout the whole body, the whole system. So we say that's systemic. It is specific and it has memory, right? So that our immune cells remember, oh, I've seen this antigen maybe 20 years ago, but I've seen that before, right? So they're able to remember, recognize, and go start an immune response against that particular antigen. And again, there's two separate overlapping arms. So two separate but overlapping arms, the humoral, the antibody-mediated, and the cellular, the cell-mediated immunity. So two parts to this puzzle here, and two specific adapted defense mechanisms here that have these qualities: systemic, specific, and memory. Some vocabulary, right? The idea of an antigen, right? So antigens are substances that can mobilize the adaptive defenses and provoke an immune response. So substances that can mobilize the adaptive defenses, so something that can trigger that immune response and cause all that chaos, the inflammation, all that mess, to address that particular issue at the moment. So antigens. Oh, if you think back to blood, we talked about antigens that were spontaneous. So the A antigen on blood cells, on the red blood cells, the B antigens, right? These are antigens, they're little surface markers that our body just normally has. So those are self-antigens. In this chapter, we're looking at antigens as being something foreign, little markers that are found on bacteria or on viruses or on fungi, on protists, on worms, these kind of things that enter the body, that our body recognizes them as foreign, these foreign antigens. So most antigens are large proteins, complex molecules not normally found in the body. So non-self antigens. So if you've seen pictures of the coronavirus, right, why do they call it corona? Corona means crown. Well, it's got all of these little things on the surface of it, right? All of these little things are protein antigens, right? So that is going to really cause problems, but our immune system will see this, right? Our immune system, as long as it's exposed, once it hides in our cell, then it becomes more difficult. But if it's exposed out in circulation, these foreign antigens will trigger an immune response. So complete antigens. Complete antigen, as the name implies, this is the entire antigen, right? The entire antigen will have important functional properties, right? And students struggle with these two concepts, right? In red here, we talk about immunogenicity and reactivity. So immunogenicity and reactivity. So in the process of dealing with, of finding, of recognizing complete antigens, if the antigen itself is immunogenic, that means it has the ability to stimulate proliferation of specific lymphocytes and antibodies. So in English, our immune system is able to recognize it, recruit more help. Let's get these white blood cells reproducing and start making these specific antibodies. So immunogenicity means that it triggers an immune response in the body. Reactivity, so the ability to react with the products of activated lymphocytes and antibodies released. So if something is a complete antigen, it can wake up the immune system, and the components of the immune system can then go chemically interact, chemically combine, chemically connect, react with that antigen. So we don't really, as a patient, as a normal human, we don't really consider this. We know that, yes, our body was able to fight stuff off because we didn't get sick, or no, our body wasn't able to fight stuff off and we did get sick, right? So, but at the chemical level, at the cellular level, these immunologists define these two terms, immunogenicity and reactivity, right? So what are some examples of complete antigens? Things like large proteins, the most common, complicated carbohydrates, lipids, and foreign nucleic acids. So all of these will trigger proliferation. So they are immunogenic and they are also reactive to the components that our body has released. So antigenic determinants. Antigenic determinants are part of the antigen. So certain parts of the entire of the complete antigen that are immunogenic. So antibodies and lymphocytes receptors bind to these, right? So yes, the entire protein, the entire antigen is foreign. There's going to be a lot of parts that are sort of hidden, not exposed to our immune system. So then we deal with these little antigenic determinants here. So here in red, right, we have this big red blob thing, circular with little components. So yes, this whole thing is an antigen, it's foreign. But on the surface of that antigen, there's little different antigenic determinants. Here we have like a little triangle, a little curved one, little like, little battery shaped one there, right? So the different antigenic determinants will interact with different types of antibodies. That's important because that means that we have various, more than one type of antibody attacking a particular antigen. So that antigen is being attacked by different antibodies at different locations. So if this antigen, or I should say, if this antibody, this little blue mess here, antibody A, if antibody A is weak or deficient, that's okay, because antibody C and antibody B might come in and help break up this antigen. So the more determinants it has, the easier it would be for our different types of antibodies to find it, attack it, and just get rid of this as quickly as possible. But this is the idea, the antigenic determinants. So that is what will enable the idea of reactivity. So where does the antibody connect to the antigen? At the determinant area, the antigenic determinant area. So that's where we have that chemistry going on. We have that chemical interaction, that reactivity, and that's where we start trying to work on destroying, damaging, getting rid of the antigen. So antigenic determinants, most naturally occurring antigens have numerous antigenic determinants. I kind of mentioned that. They're able to mobilize several different lymphocyte populations, form different kinds of antibodies against it. Now, something that is a little bit important, maybe something to consider. The more diverse, the more large, the more complicated the antigen is, the more determinants it will have. But let's say that we have something large, very chemically simple, just repeating the same thing over and over. Things like plastics, things like stainless steel, these have very little or no immunogenicity. So they don't really trigger an immune response. And if they do trigger an immune response, there's very little parts on that big protein. There's very few determinants to cause any type of reaction. Now, why is this useful to know? If you're having a bad day, right? You have a bad injury, you have surgery. Here we have this big metal plate that's been, put in. It's metal screws, right? So a lot of these simple molecules, yeah, they're big molecules, but they're not chemically diverse. Right? They're very simple. The chemical structure, the molecular structure of those is very simple. Things like stainless steel, these artificial plastics, certain types of, let's say, like stitching for lacerations. They're going to be put in the body, maybe jewelry, good stuff, not the cheap jewelry, but the good quality jewelry. You put it in the body, and basically it's not going to cause any type of allergenic reaction, inflammation. It's not going to turn the cheap necklace, turns your neck green, something like that. We don't deal with that, right? These are high quality stainless steel type of substances. So we put them in the body, and that would be not good. That would be a sort of a negative aspect if our immune system started to attack those pins and those plates. It would defeat the point of putting them, yeah? So I got this picture, a dog, some sort of dog surgery. See a big plate, a lot of stitching, and those, the internal plates can persist the life of that organism, right? They're not going to cause any issues because they don't generate any immunogenicity, or very, very minimal immunogenicity and very minimal reactivity as well. So two qualities that are necessary to mount an immune response. Without immunogenicity, reactivity, there's no immune response generated. All right, so I hope that's making sense up to that point. The next bit is a little bit different. We're still talking about antigens, but now we're talking about incomplete antigens. So not the complete antigen, we're talking about haptens. Haptens, sometimes I like to call them halftons. They're not complete, they're little halves, they're little parts of a big thing, kind of like a little determinant that basically will trigger some immune response. So they typically